So today I'm going to go over how to do a plasmid mini prep. The idea behind a plasmid mini prep is to isolate particular plasmid and purify it. So picking up from the last video where I had done the bacterial transformation, I have my grown bacterial plates. Let's see if it shows up. This is my ligation plate. So you can see on this side it's a thick field. That was the 10x side. On this side are individual little colonies. Now what I did yesterday was I picked 5 of those individual colonies and submerged them each into a falcon tube with 5 mils of LB broth with carbonicillin as the antibiotic. Then what I did was I used a single pipette tip to pluck the colony off the plate and drop it into the broth. I let that grow overnight at 37 degrees shaking, and you can see they're all very cloudy solutions. That's how you know you have a lot of bacteria in there. So to begin the mini prep, I've transferred 2 milliliters of my bacterial stocks into centrifuge tubes. What I'm going to do is centrifuge each of these tubes at like 12 to 13,000 G for a minute. That'll pellet my cells down and I can remove the media. After that, we'll come back and start the actual cell lysing. The cells have pelleted down now. Hopefully you can tell the difference. Can you see the little pellet at the bottom? And also the media is now clear. So if you compare the two, this one has not been spun down. This one has. Much more clear. Now we no longer need the media, so I'm going to pour that off into a waste container. Don't be worried, the pellet should stay in place. I've never had it fall out. And then I'm going to take a Kim wipe and just collect that excess media. You want to get as much out as you can. I'll do that for all 5 of my colony clones. Now it's time to start lysing the cells, which means to basically spill them open, break their cell membrane down, spill out the interior of the cell. So begin by using what, I don't know if I mentioned, but this whole process follows actually a commercial kit. It's called ZR Plasmid Mini Prep Kit. Typically we use the classic form. It's easier, it only has 3 buffers during the cell lysis phase, and they're color coded. So you begin by using the number 1 buffer. It's this red buffer. I'm going to add 200 microliters of the P1 buffer to each of my samples. Now, if you're careful, as long as you don't touch the tip of your pipette to any of your Eppendorf tubes, you can reuse the tip. So I pull up 200, kind of carefully hover over. I'll try to let you guys so you can see this. Might be out of frame a little bit, but basically what I do is I just hover over, fire it in. And I go right down the line. I'm going to set it down this way. Now that I've added a P1 buffer to all of my samples, I need to bring the cells back up into solution. So bring them from a pellet to a suspension. And to do that, we have, there's a vortex right here. I moved it to my bench just for the ease of the video, but we have multiple vortexes around the lab. And what they do is they shape your samples rapidly. And you'll see when the pellet has worked up, you won't see chunks anymore. And your pink solution, which was clear, should now be cloudy. So this one has the cells in it, or cells in solution. This one has the cell pellets. Okay, so my cells are up into solution now. The next step is to add 200 microliters of the number 2 buffer, this blue one. This will begin the cell lysing. Just like before, if you're careful, you can use one tip. And I'm going to pipette 200 microliters of the green buffer to my tubes. Now, this next step takes a little bit of technique where I need to mix these, but I don't want to be too aggressive doing it. So all you do is just invert the tubes 4 to 6 times to get a nice mixture. If you're too aggressive, you can spill out the chromosomal DNA, and that will contaminate your purified DNA elution. Our goal is to just isolate plasmid DNA. Now that it's mixed, you just let it sit on the table for about 1.5 to 2 minutes. And then I'll come back after that point and we will neutralize the solution. To neutralize the solution, we're going to use the 3rd buffer, which is this yellow one. This is stored in the 4-degree fridge in our lab, and I'm going to add 400 microliters of the neutralizing buffer to my cells. Now, this one, let's see what else shows up. You can see the tubes are actually multicolored right now. There's a red band, kind of a little bit of blue, and then the yellow. We need to mix that in so that it's all yellow. And you do that just by giving it kind of flicks of the wrist. You don't need to vortex it, but just enough so that it becomes totally yellow. It's difficult to see on the camera probably, but the solutions are actually really cloudy now. There's a lot of debris, like chunky kind of debris. What that's doing is it's bringing down all the cell debris, and our DNA will remain in the solution. So now that I've mixed it up and I don't see any more red, I'm gonna let it sit for about 2 minutes while the buffer neutralizes. The samples have sat on the bench top for about 2 minutes now, and they're still cloudy, but I'm trying to get the camera to focus. There's a blue debris starting to form on the top of the solution. If I rock it around, you can see that blue band a little bit. So what I'm going to do now is I'm going to centrifuge all 5 of my samples for about 4 minutes at 16,000 G. This will bring down the cell debris, and we will be able to pull off the supernatant containing our DNA. So I'll come back after the centrifuge step is done. After centrifuging, you'll notice that there's a blue debris on the bottom of the tubes now. There we go, nice and focused. So you see that blue debris kind of at the bottom slanted upwards? That's all the cell material that we don't want. So my DNA is in that clear yellow supernatant. Now, to isolate the DNA, I prepared a trap tube and a column that has a membrane which will stop DNA. So we will centrifuge that yellow supernatant over this. Everything we don't want will flow through while the DNA is caught on that white membrane. So I'm going to transfer 700 microliters of my cell supernatant onto each of these columns. And I've labeled them so that I know which one goes to which column. When you pipette this out, basically I keep it slanted and I do my best not to disturb that blue pellet. So I pulled up 700 and all that remains now really, boy this camera doesn't like to focus, is that blue pellet on the bottom. I'm going to go centrifuge these spin traps, pull the yellow solution through, stick my DNA to the membrane, about 30 seconds at 12,000 G. I'll come back after that. So I've centrifuged the yellow supernatant through the trap column, and you can see all the yellow solutions now caught in the bottom. So I no longer need that. My DNA is stuck on this white membrane. So I'm going to pour off the flow through to my samples. Now, the next step can vary depending on the cell line you're working with. What you would do is you would use a wash buffer called endowash. It removes endonucleases. However, particular cell lines can be modified to not have endonucleases. The NEB5 alpha cells that I'm working with happen to not have endonuclease. So I don't need to do the endo wash step. But it doesn't hurt if you do. I'm going to instead move on to the plasmid wash step. So there's this larger bottle that just says plasmid wash buffer on it. And I'm going to add 400 microliters of this to each of my columns. You'll notice, maybe not with a mask on, but in normal times, that it's got a strong smell of alcohol. It's a high percentage of ethanol in this. That helps to wash the plasmid. So I'm just going to transfer 400 microliters into each of my columns. And I'm going to go spin or centrifuge the columns for 1 minute at over 12,000 G. I probably won't come back for the second step, which is basically I'll take the tubes out of the centrifuge, pour off the ethanol that's now caught in the trap column, and I'll just spin again for 2 minutes to really dry it down. You want to remove that ethanol after you've washed it through. So we'll come back after that part. Alright, we're on to the final stage. So I've washed through my plasmid wash buffer, decanted it, and then I put the empty column back into the trap tube and span it again. This way I really pulled through whatever ethanol was remaining. Now, in the meantime, I got just some centrifuge tubes, labeled the caps, and I'm going to transfer the column, which has my DNA, into the centrifuge tube. It just sits inside like that. And to each of these columns, I'm going to add 30 microliters of DNA elution buffer. It's the final buffer we'll use. After I've added the elution buffer, I'm going to let it sit on the membrane for about a minute, and then a centrifuge for 1 minute at 12,000 G. And at that point, we should have isolated my plasmid DNA. You can confirm this. You can measure the concentration through a nanodrop, and then confirm the plasmid identity through a confirmation restriction digest, and put that over a gel, which is probably what I'll do tomorrow. So even though it's the same buffer, I'm actually touching the membrane with it, so I'm changing the tip on each time. So I'll let this sit for about a minute, then I'll go spin it down, and at that point I'll be done. So mini preps are actually pretty easy. They take about 20 minutes in total. That's that.